So thank you all for attending today's webinar, and as Sarah just mentioned, thanks for everyone in Europe joining us this afternoon, and good morning in the US, and for some of you that are further away from Minneapolis, it might be a very early morning, so thank you everybody for joining us this morning. So I'm going to talk today about our recombinant antibody technology that we've been developing over the last 2 to 3 years, and it will include data on actually 4 species. I'm not going to go into excruciating detail on every single piece. I'd rather have a broad overview over our capabilities, over the technologies we've developed, to actually spur some thinking on what you can do once you have isolated the sequences from various types of antibodies and the possibilities that come with that once you have a recombinant protein. So with that, I'd like to start, and I'm just going to go over the agenda very briefly. So I'm going to start by introducing our line of rabbit monoclonals that we've made recombinantly in the last 2 to 3 years. From there, I'm going to go to our hybridoma conversions, so essentially going from a hybridoma cell line-based system to more of a recombinant antibody-based system. And then I'd also like to introduce our GOAT polyclonal to mono conversions. This is more recent work, and again, I'm just going to show some data on it for some conversations on the technology. From then on, I'm going to go into, rather than talking about full-length IgG type molecules, on the modifications. Now that we've isolated the sequences, I'd like to go into all the possibilities that come with recombinant modifications and molecular biology techniques. So I'm going to introduce some data on FABs, Fab'2s we've made that way, isotype changes, other Fc modifications that are relevant for flow cytometry, and abolishing some non-specific binding and whole cell work especially, as well as some of our capabilities on custom services, if that's of interest to you. So before I start going into details on the recombinant system, I always go back and start on the classic systems, which is hybridoma monoclonals, which is where we started obviously 25 years ago. So we have a large inventory on hybridoma monoclonals. We've always developed polyclonals alongside them, and then in recent years we've actually introduced more recombinant monoclonal type based systems, the rabbit being the predominant species for that. However, once we went from hybridoma systems into a conversion system. So if you already have cloning capabilities, then you can actually take your cell line banks, which is what we did, and you can make a hybridoma antibody into a recombinant antibody. We also developed a system that allows us to then convert the polyclonals into a recombinant antibody, and once you've done either one of these approaches, what you get is actually a sequence-verified antibody, as well as high lot-to-lot consistency, which has been talked about in recent years in the literature to a large extent. Once you have it sequenced, again, you can do Fc modifications, you can add tags, you can isotype swap. So you have a lot of possibilities open to you that you're used to from standard recombinant systems on proteins in general. So the one thing that we came across in the last year that I just wanted to bring up here first is there seems to be 2 misconceptions sometimes out there within our customer base. And the first one that I just wanted to address very briefly is that recombinant antibodies do not require animals. While that can be true for some of the phage display systems that do not use animal-based libraries to begin with, other labs have chosen to go with a pre-immunized library, so they actually do start in an animal, they pre-immunize the animal with the target of choice, and then they go ahead and build their libraries based on that. Again, the animal work in that case is minimal, but it does require an immunized animal in some cases. The other system that people have been using for recombinant antibodies is cloning directly from B cells. And in that system, definitely you will start with an immunized animal. Very rarely will you ever start with a naive animal. I haven't seen anything along those lines. So cloning from B cells definitely requires immunized animals. The second one that we've run into with customers when we talk to them is there is this perception that all rabbit monoclonals are hybridoma-based antibodies. Again, that is not correct, as the B-cell cloning process that I'm referring to under the first point can also be used for the rabbit monoclonal recombinant system, and that's what we've actually employed in-house. So that is direct cloning from B cells, which is by now an established technology. Obviously, the second system is utilizing a fusion partner for rabbit B cells. However, rabbit hybridomas are more notoriously unstable, and it's been described in the literature as well if you'd like to find out more about that. So based on these two, we have chosen in-house to go with a system that actually utilizes B-cell cloning. So this is the overview on a high level of what our process looks like. We're utilizing this to make any recombinant antibody from our B cells. So we're starting with an animal, we're immunizing it, we're boosting it, same as you would with a standard fusion-based mouse monoclonal. At that point, we're isolating PBMCs, and then we're cloning out of the PBMCs, we're isolating and making a small-scale expression. Small-scale expression then gets screened on applications, and our applications range from Western blot to flow, or the traditional standard antibody applications people would look at, Western ICC, as well as all the way out to our ELISA applications. We're then screening the clones that have been picked by a group of researchers that have reviewed all the data for specificity, sensitivity, and we're picking the best clones based on the panels. Those get sequenced, they go to a pilot scale, they go to purifications. Again, all application testing is repeated at that point to make sure that what we picked initially is still the same result that we're obtaining the second time around. So we're building the validation and the robustness right into the development system for our antibodies from the beginning. So I'm going to show you a couple examples of what you can expect from our rabbit monoclonal antibodies using this development process. And we're obviously known for our cytokine proteins, for our cytokine-based antibodies, chemokines, and those types. So today I actually wanted to use this as an opportunity to present data to you on the targets that we might be less known for, and one of the areas we've spent some work and effort on were actually anti-tag specific recombinant rabbit monoclonals. So I'm presenting you data today on 3 of them. One is anti-V5, anti-FLAG, and anti-HA. And the reason why we focused on this particular line of targets is there's a lot of anti-tag antibodies that will only recognize tags that are either N or C terminal. So we decided we wanted to make sure that the anti-tag antibodies we're putting out there will recognize both N and C terminally tagged recombinant proteins on a Western blot. We've had a lot of customers that say I have to buy 2 because one only works for one well but not for the other. So we wanted to address that right away within the design of the antibodies. So for these 3, we've clearly met that specification. In addition, we used BioCore to actually measure the affinities, and I've got the results below on the below the Western blots in the graph. So our anti-FLAG antibody actually has an affinity of 6.6 nanomolar, while our anti-HA antibody is at an affinity of about 4 nanomolar. So these are high affinity antibodies working really cleanly in Westerns. We've also done flow and ICC using these particular antibodies as a secondary type of detection system. In addition, I wanted to bring some data that is more a more classic type of target line for rabbit monoclonals. So the rabbit immune system is able to recognize small haptens better than a mouse system. That has been also described in the literature over years, and it's a known phenomenon on the rabbit immune system. So a lot of people taking advantage of that and making rabbit antibodies towards phosphorylated targets. And again, we built a whole line of phosphorylation antibodies in the last 2 years as well, and I wanted to show you 2 examples from that particular target line. So the first one is our anti-REL-A This is the phosphorylation site 563 that we were targeting. And on the left, I'm showing you the Western blot on HeLa cells. The HeLa cells were either untreated on the very left or in the second lane treated with TNF-alpha and anticalyculin, triggering the phosphorylation of the target at that particular phosphorylation site. And as you can see, upon treatment of the cells, you can actually detect a band expected at the expected molecular weight of about 85 kilodalton. We did the same treatment, different cell type in ICC, and again you see the increase in phosphorylation and the increase in signal very nicely on the right panel versus the left panel. The left one being the untreated, the right panel being the treated HT29 cells, and you see a very nice signal in the cytoplasm upon treatment with TNF-alpha and calyculin in those cells. The second target I wanted to show you is the anti-risk. This is phosphorylation site serine 380. And again, the Western blot on the left shows you Jurkats. The Western blot on the right is HeLas, treated and untreated. And upon treatment, you clearly see the uptake in phosphorylation, and you start detecting the band at about 93 kilodalton, where it is expected for phosphorylated risk 1. We used the exact same cell model in this case in ICC, and again, upon treatment of HeLas with PMA, you see the clear phosphorylation signal increase on the left side versus the untreated HeLas on the right. We're also showing you the control, which is just the DAPI channel. So once we've identified clones, it's not just good enough that you do it once, so I just wanted to go over very briefly how we make sure that every single time you receive a product from us, it maintains that same high standard of validation quality and performance. So we've built this in, again, during the development phase, all applications are first established. We're then going into a manufacturing phase where all applications are tested one more time to make sure it's absolutely reproducible. There is no shift between the development lot and the manufacturing lot. Then we're packaging the antibody, and we just want to make sure that nothing happened during packaging on these antibodies. So again, all applications tested in addition on our bottle material before it goes out to you as our customers. When we make a second lot or any higher lot after that, again, we're doing the same testing scheme. So we make a large lot, we make a bulk lot. All applications are again tested on that bulk lot compared to the previous lot, and the process repeats itself again on the bottled lot from that second bulk lot before it goes out to customers. So that's how we've built in the robustness of making sure that lot-to-lot variations are caught in-house and don't even make it outside. So let me switch gears. So we had the technology established clearly for rabbit monoclonals, and we looked at our capabilities and said, we can actually apply this now to our rodent banks. And that was about at the same time when the Nature paper came out that actually called for researchers should use recombinant antibodies or binding reagents if they are obviously available. So we looked at that and we said, we can actually do this. We went back to our Hybridoma banks, we optimized our cloning, and we have now converted over 100 of our mouse and rat hybridomas to a recombinant version. When we do this, we maintain the original isotype, so we decided to go with the approach that the recombinant version should very closely mimic the original clone, because that's what customers are used to and we didn't want to change that. So that obviously cost a little bit more work on our end, but it was absolutely worth the investment on our side. The antibodies that we've cloned span all applications, so we maintain all the original applications between the hybridoma and the converted recombinant antibody. During this process, we actually improved yields through the recombinant expression system that we have established, and at the same time we were able to resolve some of our manufacturing issues on some more difficult clones, and I'll show you one example. In this case, we had to go through an isotype change, and again, I'm going to show you one example for that. And by now, we've even done some custom work for our pharma or diagnostic partners. So this is an example of a typical result obtained on the left. So this was our anti-human synuclein gamma antibody. This was a mouse monoclonal based on a hybridoma cell line going in. We cloned out the genes, we put it in our expression vector, and on the left is the Western blot as a comparison between the hybridoma and the recombinant monoclonal. They give the exact same result. They detect the same band. There is no increase in non-specific detection of any other proteins. And on the right are the ICC and IHC results for the same recombinant antibodies. Again, we're maintaining the original isotype in this case. We're maintaining the original applications. So the end result is that the recombinant antibody is indistinguishable from the hybridoma parent. The only advantage, or the advantage of that is that we know the exact sequence of this antibody. So going through this for the next, let's say, 10 years, we sell the same antibody for 10 years, we can always refer back to the parent cell line's sequence. And if anything ever should shift, it would be very, very obvious because now we've got the exact amino acid sequence for these antibodies. The other question we have gotten from our customers is, can you please show us that the affinities haven't shifted between the hybridoma and the recombinant? So based on those requests, we went back and we actually did BioCore experiments. On the left is the hybridoma. This is for our anti-TNF-alpha antibody. And the hybridoma has a binding affinity of 3.4 picomolar, while the recombinant version has a 6.4. Again, they're in the same ballpark of the affinity, so we really haven't changed anything, staying with the exact same backbone and the exact same isotype. Another example is our PARC7. Again, the KDs are staying in the same ballpark. One has a 3 picomolar for the recombinant. The hybridoma is at about 4, 4.5. They have about the same on and off rates. So there's no intrinsic change going from a hybridoma-derived antibody to our recombinant-derived antibody, except that now you know we have the sequence in-house and we have no shift in that particular product. So once we have all these antibodies cloned, obviously we need to make sure that we can manufacture these in the long term. So we optimized our expression systems to the point where we can obtain gram levels in our bioreactor systems. In some cases, traditional hybridomas, they would require about 5 to 10 times more media volume. They would require a lot more work, a lot more purification time. So we actually reduced the time for us to manufacture these for the same amount of antibody in the end. We've optimized our expression vector designs along the lines to get to this point. We have our own in-house media developed alongside this, so it's really one system co-optimized at every single step. And on the lower right, we have an example for one of our antibodies where we made 3 lots. And as you can see, within a small variance, we are getting the same yields over and over again with the same applications, the same specificities. As I said in the beginning, is sometimes we're obviously having hybridomas that are notoriously difficult. They just don't express the antibody of interest. A lot of them are linked to IgG3s. So I'm just going to show you here one example is on the top right for the pilot scale. The original wild type was at about 5 micrograms per mil or 5 mg per liter. Obviously a very low producing antibody, but we did maintain it for years. It is a very good antibody specificity-wise, but it takes us a lot of time to make the material. So we took the clone, we sequenced it, and we made some variances on the backbone of this one. And based on that, we chose, I'm just going to show you 4 clones here in pilot experiments, which already showed much greater expression levels than the original wild type. We then took one of them, in this case we chose number 5, and we scaled it up to a 3-liter bioreactor. And as you can see, while the wild type has the very low expression as we've seen for years, the cloned version now actually gives us close to 1.5 grams in 3 liters. So we're way beyond of where the wild type started. So we've gone with this new clone to actually improve our production method. And again, the applications have been maintained, and as you can see on the left, the ELISA results in this case are indistinguishable between the engineered version and the native form. The other species we started tackling was goat polyclonals. So we knew we could do the cloning successfully for rabbit monoclonals. We had the system working for mouse and rat, going from Hybridoma. So we went back and said, okay, we have all these cloning vectors now developed. Can we actually go into our goat polyclonal line and use the same process that we're using pretty much for the rabbit monoclonal, starting with an immunized animal and then isolating a monoclonal from there? Why would we do that? Obviously, you get much higher lot-to-lot consistency. So you don't have the variation in the antibody between the lots, since it's now a sequenced version and it's based on one vector. We wouldn't need to re-immunize new animals if we ever run out of serum or when the animal eventually dies of old age. In general, there is a greater demand now for monoclonals. We are slowly moving away from polyclonals, obviously, for reasons, for the reproducibility reasons that have been mentioned in a lot of the publications recently. And it's also a custom manufacturing opportunity if anybody is interested in that. So the process looks exactly the same as what we're using for the rabbit monoclonals. Again, we're isolating the PBMCs from our immunized goats. We're cloning them. We're doing a small-scale expression to make sure that the clones are meeting our specifications, our sensitivities. We're screening them in the original application, and then we're going into pilot-scale purification, and we're repeating the testing again before we release these. So one of our most recent examples is our goat anti-human MMP1. It gives beautiful IHC staining. We've actually been able to get much cleaner staining with the monoclonal, goat monoclonal, compared to its polyparent, showing here cytoplasmic staining in human ovary on a paraffin block. The Western blot signal on the left is lower. So the reason why I wanted to show this particular one is that when you're going from a polyclonal to a monoclonal, now your monoclonal only recognizes a single antigen on the target, while the polyclonal obviously will recognize multiple ones. And that will drive sensitivity on a Western blot. It can also drive unspecific binding, but the polyclonal in this case was clean to begin with. All our polyclonals are affinity purified. So the monoclonal actually will not quite meet the sensitivity compared to the polyclonal. So food for thought is we can isolate monoclonals from polyclonals. The process is very stable. Is it always the better option? Is more a question for customers. When you choose an antibody, polyclonals can have a higher sensitivity in Western blot and assay setups. They in general give very good Western blot antibodies if you're careful in selecting your polyclonals. But obviously the lot-to-lot variation is much more difficult to control. So we're going to go and offer more goat monos and polys combined, and they all have to meet our same standards going forward. So now that we have a large inventory of full-length IgGs cloned, we decided what else can we do with all the sequences, how many other modifications would we want to tackle. So I've already covered pretty much the top part of this graph, so we went into the modification of the FC regions. So obviously once you have the FC region cloned, you can add tags, you can switch isotypes, you can switch FC species, so you can swap a rabbit FC constant region, for example, for a mouse FC region, where you can introduce specific modifications and point mutations which can make for better flow cytometry reagents. since you're abolishing some of the receptor interactions. And then the other part that we've addressed is we've always wanted to make FABs in a much more consistent way. A lot of those reagents have been used in flow cytometry and neutralizing experiments as well as in vivo imaging. So those were the two parts that we've been working on for the last year. So the isotype switch, I have one example here. We started with an IgG3. Again, IgG3s are a little bit more difficult in general to maintain as a hybridoma cell line. And these are small-scale expressions, and the concentration we were getting from small scale was in the range of 7 micrograms per mil. So we swapped the backbone from an IgG3 to a G1, 2A, and 2B, and depending on the backbone, we were able to actually increase the expression level up to 5-fold. So the 2A was in the range of 40 micrograms per mil. Again, these are small scales. This is before scale-up. We then ran the Western blot against HUVEC as well as our recombinant arsenal of proteins. So we ran this against recombinant human MAPK11, 12, 13, and 14 to ensure specificity within the family. And you can see on the lower part of this slide that all the changes still work well in Western blot. So the isotype swap in this case gave us higher yields but maintained the sensitivity and specificity. So then we started addressing our project of making FABs and FAB'2s in recombinant. We've gotten a lot of customer requests over the years for FABs, FAB'2s. The traditional way is making these by enzymatic digestion, and any of you who have ever tried to do that know how difficult and daunting that can be. You have tremendous losses. It's never really an absolutely clean result at the end, and it can be highly frustrating. So we've eliminated the enzymatic method, going in a recombinant way. We were getting higher yields in the end. Going from an IgG to an FAB'2 or an FAB will eliminate non-specific Fc interactions with receptors and whole cells especially. So I show you some examples where this leads to lower background. In addition, you can add tags to these for easier detection in any application downstream. So these are 4 examples that we've recently cloned from a full-length IgG going into an FAB'2. And each one of these we introduced a stop codon to either make an FAB or an FAB'2. In addition, it has an affinity tag, that's the green dot, to be able to detect it afterwards as well as help with some of the purification. So on the right is our SDS page gel of those particular 4 small-scale lots, and you can see that the result is very clean. It gives you 1 heavy chain, 1 light chain band. You have obviously no residual IgGs since you've already eliminated that by design. It's one of the most common encountered problems if you go enzymatic. And under non-reducing conditions, you see 1 clean band for the FAB'2 at the expected molecular weight of 110 kilodalton. So then we went ahead, this is another target we worked with, and we actually did the Westerns on this. We wanted to see how well would they perform in a Western, and for this particular result we were utilizing our new technology called Simple Western. So we ran recombinant human IL-6 on the capillaries under reducing and non-reducing conditions. If you don't reduce human IL-6, you will encounter dimers. That's why we did that, and I've indicated that on the picture here. And then we came back and we either detected that with the FAB or with the FAB'2, and as a secondary we used either an anti-tag HRP or we used an anti-mouse kappa HRP. For this particular example, the anti-mouse kappa HRP gives you better sensitivity as you can attack more sites on the primary antibody, and it gives you better sensitivity on the gel. So you can actually see the human IL-6 dimer, while with the anti-tag antibody it's a little bit more difficult to get that sensitivity. But overall, both the FAB and the FAB'2 perform very similarly to the whole IgG molecule, and as expected you lose some sensitivity going from a whole IgG to an FAB or an FAB'2 since you're truncating the Fc region. So on the left we have human TNF-alpha. Again, it's the same assay system that we've utilized before using alkaline phosphatase transcription reporter assays. And the full-length antibody in this case had an IC50 of half a nanomolar. When we're changing it to an FAB'2, that IC50 goes up to 1.4. And if you're truncating it even more, now you have a single binding site for the FAB, it actually goes up to a 2.5. That one is an expected result since you're only interacting a single antigen binding site now with the target, and your blocking activity does decrease with the size of the molecule. The human interferon gamma neutralization assay resulted in a different outcome, where the full length and the FAB were behaving fairly similarly, but the FAB'2 actually gave us lower IC50s. So in this case, the truncating the Fc region might result in a change of the folding of the antigen binding site. It might actually work to our advantage. Again, that's a working theory. We're still investigating some of these results. I just wanted to show you the different results you can expect going from a full-length antibody to an FAB'2 and then to an FAB, that it does vary with the target. The other assay we ran on the FABs that I've shown you in the gel before, one of which was CCR7, was flow cytometry. CCR7 is a marker employed on T cells. And we ran the full-length IgG controls on the top, and then we came back with a goat anti-mouse APC secondary. So you will get a little bit more background potentially using a secondary system, but we wanted to make it a very comparable test. So the top result shows you the IgG control on the top right graph, and you get a very nice population of 33% detected in PBMCs. When you're going to the FAB'2, and in this case we detected it with a goat anti-mouse APC. This is a generic goat anti-mouse. It's not the Kappa version we used in the Western blot. You still pull out your T cell population, but the separation between the labeled cells and the unlabeled cells isn't quite as clear. However, if you use the anti-tag APC, again, you get a very nice separation. You get your specific T cell population with a very low background. So the antibody still performs very well as a FAB'2, just removing the Fc region. Another example is CD3. Again, you get the same result. In this case, there was really no distinguishing features between using a goat anti-mouse PE versus an anti-tag PE. They performed exactly the same as the original full-length IgG. So now that we had more of an idea of how the FCs interact, especially in whole cells, we tackled the next problem that sometimes you will see high binding in flow cytometry between the FC region of your antibody as well as the receptors on the cells. Normally this is being addressed obviously by using FC block. That's the standard procedure in flow cytometry, but we went one step further and we said, okay, we would actually like to investigate a little bit more on which mutations could we introduce into the FC region to abolish some of those interactions between cells and the detection antibody. So we made a bunch of mutations indicated on the top. So on the top of this graph, we just call them mutation 1 through 2, 3. Those are single mutations introduced. And in addition, we made the dual mutation 1 and 2 combined, as well as all 3 mutations in one FC backbone. On the very left is the result of the original parent, the unmodified recombinant version. And as you can see, if you focus on quadrant 1, it gives you a high non-specific binding for this particular antibody that we were running. The mutations 1, 2, or 3 alone did not abolish that interaction, and you still see a large amount of non-specific binding on the lymphocytes as well as on the monocytes. And using mutation 3, it actually got worse, so that was just not the way to go, obviously. But when we combined mutation 1 and 2, that was the one that actually abolished a lot of the non-specific binding and gave us still our expected specific binding and pulling out the population as that was our final goal. So in this case, we know how we need to modify this particular antibody to actually make it a better flow reagent, and those are modifications we might introduce going forward into some of our flow cytometry-based antibodies to make sure that customers don't have as many problems with non-specific binding to FC receptors. So what does the recombinant antibody workflow look like? There's multiple points where we can start either switching between a hybridoma or a polyclonal, going into a recombinant system, as well as there's multiple steps for customers if they're interested in specific projects. So we can either start with an immunized animal. It's either an internal project, could be a customer-driven project. We go through a hybridoma process, or we go through a B-cell isolation for rabbit monoclonals, and then we go into our clone construct pathway and make it a recombinant conversion at that point. In some cases, customers know exactly which sequence they would like us to clone, and we go straight from sequence into cloning expression. We send it then back to customers, and they can test it in their own applications. In other cases, we have large hybridoma banks. Customers have screened our hybridoma banks for their particular applications before, but then they decided, okay, we don't want to have this going forward as a hybridoma product. We would prefer to have it as a recombinant. We would really like to know the sequence, or they want to introduce small modifications on the FC region especially, and then we'll convert it again in that case from the hybridoma that they originally picked into a clone construct. It works the same as if they were to provide us with a hybridoma from their own bank. So it's a very, very flexible process, and a lot of it can be done using molecular biology tools. And then we take it all the way to production, and again at that point it goes back to either our own inventory or it goes back to the customer for final approval. So in short, we're now offering everything from de novo antibody development from standard classic hybridomas all the way to our recombinant rabbit, mouse, rat monoclonal lines. I hope I've shown enough data to convince you we can convert goat polyclonals to monoclonals. We can do a lot of FC modifications. We can do isotype changes. We've done species swap, which I haven't shown you data for today. We can add tags. Again, we have a lot more data on that. We had to be a little bit more selective on what we're showing today, or it would get too long. And we can do a lot of custom work using all our tools in-house. And with that, I would like to thank you, and I welcome any questions that you might have.
Thank you very much, Birte. That was an excellent presentation. We have quite a few people who've asked some questions.
Okay.
So let me just... So we have one from Rose Mage, which is similar to quite a few that other people have asked. So I'll just unmute you, Rose. Can you hear me, Rose?
Yes, I can hear you.
Please put your question to Birte.
Well, when you were talking about the goat polyclonals, I thought that you could possibly mimic goat polyclonals by making a defined mixture of several monoclonals selected from the original polyclonal producing goat. Have you ever tried that?
Yes, you're absolutely correct. You can do that. I haven't shown data for that. That is something we have tried in-house. Yes, you can mimic a polyclonal. The only question is how many clones would you feel comfortable with mixing reproducibly? But absolutely you can do that, and that's something we're going to investigate more going forward, most likely. You can do the same with mouse monoclonals, obviously. You can make an artificial polyclonal. You're correct.
Okay, thank you very much for your question, Rose. The next question we have from Teresa Bengtsson. So, Teresa, are you there?
Yes, I am. Hello.
Hello. Please put your question to Birte.
Yeah, I had a question about the backbone that you changed. Do you have any general idea of what backbone is increasing the production levels the most?
You're talking about the isotype swaps?
Yeah, isotype swaps, exactly.
We don't really have the same result for each antibody. We know that when we switch from an IgG3 that are problematic to begin with, we can always increase it, but I cannot tell you that it's always going to be the 2A or always going to be the 1. We're not quite sure yet what drives that. There's probably folding obviously involved between what the variable region looks like versus the constant region, but it's not 100% predictive.
Okay, thank you. But I have a follow-up on that.
Yeah.
Have you seen any, have you solved any precipitation issues with your antibodies by swapping isotype?
Not yet.
No? Okay. You have other issues then.
Well, we have precipitation issues with some of the parents, which is what's driving some of this these investigations, obviously. So we've been able to do it the reverse, where we have a precipitating clone, and swapping the background again addresses that. So that's why we're going it around this way. And then when we make all 3 or 4 versions, obviously we're monitoring those, and then we're choosing the one that, if we were to see it, we're not going to go with that. But again, we haven't done large numbers yet that I can tell you for sure. But I haven't seen it so far.
Okay.
So.
Thank you.
You're welcome.
Thank you, Teresa. And we have one more question from Ian Wilkinson. Are you there, Ian? Hello, Ian. Can you hear me?
Oh. Hi, can you hear me?
Yes, we can.
We can hear you.
So I was just wondering what expression system you use for your recombinant antibodies.
Are you asking about the cell line itself?
Yes.
Okay, we're using a HEC293 system, and I probably should have pointed that out on the FABs. I actually had it on the title. We're using our HEC system for everything that I've shown today. We're not using a CHO system.
Okay, thank you.
You're welcome.
Thank you, Ian. Let's have a quick look. I think that's probably all the questions we have for the moment. We've just had one from Dersing Lei. So I'll just go to Dersing. Dersing, I apologise if I've mispronounced your name. Are you there? Oh, no, I don't think we can get him on the speaker. But his question was, oh, hello, are you there?
Oh, hi. Yes. Yeah, I just want to know how is the glycosylation of all the clone? Are they the same or different? Will they affected about your affinity or something like that?
I'm sorry, could you repeat the question?
The glycosylation on the antibody.
Yeah.
Are they all the same, or are they have different glycosylation? Because some scientists argument glycosylation is very important for the antibodies.
Okay, so yes, glycosylation is definitely important for antibodies. I would agree with that statement. At the same time, I have to say we haven't done a study to actually in detail analyze the glycosylation patterns. We have decided to stay with one cell line expression system for that reason, the assumption being that it's going to be at least reproducible within one system. That's the other reason why we're staying with the HEC293s also for FABs, because you can see some companies or some researchers going into an E. coli-based system with FABs. Yes. And we've strayed away from that because we want to maintain the glycosylation to the best of the extent. I mean, obviously it will be different from a hybridoma system, I would expect that, but it doesn't seem to affect affinity or specificity switching from a hybridoma to a hex cell line.
Okay, so if you think about if you change to the prokaryotic system or yeast system, would that any change?
I would expect changes, but I cannot predict that it's gonna abolish function completely. It probably depends drastically on the target and what the ingoing antibody is.
Okay, great. Thank you.
Mm-hmm.
Thank you very much, Dersing. I think we're coming up to our time limit now, so if you have asked a question that we haven't been able to get to you today, we will be collecting all your questions and answering them by email, so we will get back to you. So I'd like to thank Birte very much for her time. This webinar has been recorded, so will be available for you to watch again. We'll be emailing out a link in a follow-up email to you later on in the week. So thank you very much for your time and have a great day.